Okay, good afternoon, everybody. So I'm here to tell you a little bit about Cynata Therapeutics. So we are a clinical stage biotech company based in Melbourne, Australia. The company is entirely focused on the development of a proprietary platform manufacturing technology, which we call Cymerus. And what Cymerus is essentially an iPSC-based platform for the scalable manufacture of consistent quality mesenchymal stem cell or MSC-based therapeutic products. And as you will no doubt be aware, those MSC therapies can be used to treat a wide range of serious disorders with unmet needs. So this platform is really unique in that it is an iPSC-based process, and certainly to my knowledge, all of the other commercial clinical stage MSC manufacturing processes that are in use at the moment are relying on isolating cells directly from donated tissue. And that comes with some challenges, which I'll tell you a little bit more about in a moment. So over the past number of years, we've generated strong safety and efficacy data in a range of preclinical and clinical studies, and several of those studies have been published. We now have 4 active clinical programs, which I'll tell you more about in a moment. And collectively, those indications represent a pretty large addressable market. So it's been estimated at about $28 billion annually. The company right now is well funded to complete these ongoing clinical trials, and in fact 2 out of the 4 clinical trials are funded externally, so there's really just 2 of them that we're funding ourselves. So our clinical pipeline, as I mentioned, there's 4 programs now. We have 2 that are Cynata sponsored and managed and funded, and 2 that are partnered and funded and managed externally. So the 2 Cynata-sponsored programs are graft versus host disease, which is now in phase 2, and diabetic foot ulcer, which is in phase 1. Our partnered programs include osteoarthritis, which is in phase 3, and that's partnered with the University of Sydney and funded under an Australian government NHMRC project grant, which is kind of like the Australian equivalent of the NIH. And we've just recently announced the approval of a new clinical trial in patients undergoing renal transplant, which is in partnership with a group in the Netherlands called Leiden University Medical Center. And again, they're providing the funding for that trial and they're managing it. But one important point I'd like to make is that those 2 partnered trials, although they're not being funded or sponsored by Cynata, we do have full commercial rights to use the data that arises from them. So MSCs, no doubt everybody in the room here would be fairly familiar with MSC-based therapies. They have obviously been very widely investigated by a number of companies and many different academic groups over the past, say, 10 to 15 years in particular. And the main reason is to harness their immunomodulatory and anti-inflammatory properties. I don't really have time to go into the mechanisms of action too much here, but I suppose one key point is that this is not a stem cell therapy that is intended to engraft and differentiate. These cells work largely by paracrine means. It's more of a drug-like effect where the cells release their bioactive molecules and then they're cleared from the recipient relatively quickly. So as I mentioned earlier, our platform is really unique in terms of MSC manufacturing. So a conventional manufacturing process for MSCs involves isolating the MSCs from donated tissue. For example, bone marrow, adipose tissue, placenta are probably the key ones, umbilical cord blood also. And they then take those isolated MSCs and expand the cells in culture to generate sufficient cells to treat patients. But there's a fundamental challenge here, which is that you don't get many cells out of each donation. So you might get, for example, 20,000 cells from a bone marrow donation, but you often require upwards of 1 billion cells for a single patient treatment regimen. So you can see right away that a great deal of culture expansion is required. But of course, most people aren't trying to just treat one patient. They're trying to make large allogeneic batches to treat thousands of patients. And then, of course, the level of expansion that's required is even greater. And the reason that's an issue is that MSCs have been shown to change as they are extensively expanded. So they start to lose their potency and ultimately enter senescence. So there's a limit to how many doses you can make from one donation, certainly without losing the functionality that you're trying to harness. And so what that means is you've got to go back and get new donors on an ongoing basis. But that's a challenge as well, because it's very well established that there's really substantial variability between donors. And so those two issues are really what the Cymerus process is intended to address. So by using iPSCs have the capacity to replicate themselves essentially indefinitely without perceptible changes. So we can make essentially all of the cells we'll ever need from a single cell bank, which came from a single donation from a single donor. So we completely eliminate that challenge of donor-to-donor variability. And also, because we can get the scale at the MSC stage, or sorry, the iPSC stage of the process, once we differentiate those cells into MSCs, we don't have to expand them a great deal at that point. So we also avoid those issues with excessive expansion of MSCs. And so we have a proprietary patented process that it really is, that is the core of our IP. It's the ability to differentiate the iPSCs into MSCs. Now, it's worth noting that MSCs from different sources have different properties. So we supported a PhD project at Monash University in Melbourne, which is recently completed. This project looked at MSCs from various sources, obviously Cynata's iPSC-derived MSCs, also bone marrow, adipose tissue, and umbilical cord blood-derived MSCs. And there was various comparisons, of course, across this project, looking at gene expression, protein expression, and so on. And it did find notable differences between the MSCs of different sources. It found that the iPSC-derived cells were most similar to cord blood-derived MSCs, which is perhaps not too surprising, given the iPSC MSCs are considered to be more kind of a primitive or younger MSC. But I think one of the most important findings was that it showed that there was substantially less variability between batches in the iPSC-derived MSCs, and indeed less heterogeneity within batches as well. And so that's really kind of validates the premise that we're trying to follow here. So we have a strategic manufacturing partnership with Fujifilm, which of course is a leading player in the manufacture of cell-based therapies. So they are providing contract manufacturing and development services to us, and we don't have our own manufacturing facilities, so this is obviously an important part of our value proposition. So we completed our first clinical trial in steroid-resistant graft versus host disease. If people are familiar with this disease, it's a really terrible condition, very difficult to treat, and in patients who fail to respond to steroids, the prognosis is extremely grim, with mortality approaching about 80% after 2 years. We treated 15 patients who had steroid-resistant disease, and we saw really encouraging results. So 13 out of 15 showed a response, and 8 out of 15 showed a complete response. We also saw really encouraging survival results. We saw a 60% survival rate at 2 years, which in the context of this population is really very high. It's worth noting that recently a drug, a JAK kinase inhibitor, ruxolitinib, has been approved for the treatment of steroid-resistant GVHD. And in their phase 3 trial, they didn't report 2-year survival, but they reported 18-month survival, and it was just 36%. So again, that shows that the 60% survival rate that we saw is really very encouraging. This trial was published in Nature Medicine, and I think that was recognition of the fact that it was the first completed clinical trial with iPSC-derived cells of any type in any disease. We're now in a phase 2 study in GVHD. We're pursuing a slightly different population now. We're looking at newly diagnosed GVHD patients who've been identified as being at high risk. This is a global trial which we intend to take place in Australia, Europe, and the US. We have approvals in both Australia and the US, and we have opened for recruitment in Australia and expect to open for recruitment imminently in the US, with the European centres coming on a little later. This is a 60-patient randomised controlled double-blind trial. We're expecting to complete recruitment at the end of the 2024 calendar year, with results expected in the second half of 2025. The other Cynata-sponsored trial that we've got ongoing is in diabetic foot ulcers. This is a slightly different approach where we're actually seeding the MSCs onto a novel silicone dressing and applying that to the wound. So it's basically an MSC wound dressing. This trial commenced last year, and we're seeking to recruit 30 patients in total who are randomized between standard of care and the active dressing. We're expecting recruitment to complete by the end of this calendar year, with results available in the middle of 2024 calendar year. But because this is an open label trial, we do have some initial data. So this is from the first 6 patients who were enrolled, and what this graph shows is the percentage ulcer healing over time up to 28 days. And what you can see is in the light coloured line, the top line there is the active group, and you can see that there's a substantially higher proportion of the wound has healed at 28 days. Now obviously this is a small subset of the patients so far, but these results are very encouraging, and I think if we see similar results with the full data set, this would certainly be a program that would have great promise. So our osteoarthritis trial, as I mentioned, this is partnered with the University of Sydney, and it's funded by an Australian government grant. This is a phase 3. It's a large trial, fully randomized, placebo-controlled, double-blind. It has already recruited more than 300 patients, and we're expecting recruitment to conclude around the end of this year or perhaps early next year. This has a 2-year follow-up, this trial, because we're looking for a disease-modifying endpoint. So unfortunately, you've got to follow the patients for quite a while in order to evaluate that. So the results are still a little way off, but this really could be a huge trial for us and indeed for the treatment of osteoarthritis. There are currently no approved disease-modifying agents for osteoarthritis. So if we were to show that sort of effect, it really could be a game changer for how these patients are treated. And of course, osteoarthritis is an enormous market. It's a very common condition and indeed a growing condition. And the final trial that we have about to commence is a trial in patients who are undergoing kidney transplant. And as I mentioned earlier, this is in partnership with Leiden University Medical Center in the Netherlands. The idea here is that, as I'm sure everybody would be aware, when somebody receives a kidney transplant, they then typically have to take immunosuppressant drugs for the rest of their life in order to prevent rejection of the transplanted organ. And those immunosuppressant drugs are very effective in preventing rejection, but unfortunately they come with very troublesome side effects. And in particular, the calcineurin inhibitors, which these patients typically receive, not only are associated with a high rate of serious infection risk, also an increased risk of cancer, but ironically they also cause kidney damage. And of course, in patients who've just received a kidney transplant, that is clearly the last thing you would want. So there really is a strong desire among clinicians to try and move away from those types of drugs. And this group have done a pilot study using bone marrow-derived autologous MSCs, which showed some promise. And the idea is that after the transplant, the patient receives MSC treatment. And in our trial, the calcineurin inhibitor dose will then be dramatically reduced. And the ultimate aim is to actually withdraw the calcineurin inhibitors entirely. And of course, the benefit there would be that we avoid those really troublesome adverse effects. And of course, the aim is to do that without an increase in rejection rate. So this trial received regulatory approval in August, and we're now just working on the final steps in terms of getting that trial up and running, and we hope it will commence in the very near future. So just to finish off, I've only got a minute left, so I will go through this pretty quickly. But I guess the key point is that our manufacturing process in the MSC field is really unique in terms of commercial clinical stage processes. Our iPSC-based platform allows us to make an essentially limitless number of doses from the same cell bank, which came from a single donation from a single donor, and that allows us to avoid problems with interdonor variability, and it allows us to also avoid excessively expanding the MSCs, which is associated with loss of potency and loss of functionality. We have numerous ongoing clinical trials, and we're going to have readouts at various times over the next couple of years. And I guess one thing that I didn't cover during the main presentation there is we really have a partnering-driven business model. We are aiming to out-license our assets and indeed make our platform available to partners for other applications that we're not currently exploiting. So we'd be very happy to talk to anybody who has an interest in that. Thank you very much.